Hey guys, so here we are for chapter 16. It says the molecular basis of inheritance. For the last couple of chapters we've been talking about genetics and the passing on of information and how you receive characteristics that affect the way you look, your phenotype. And we know that all comes from our genotype, the genetic information that we get from our mom and dad. But we haven't really talked about the main molecule involved in all of this, DNA. How does DNA itself copy or make copies of itself, replicate itself to pass that information on to the offspring? So this chapter is all about DNA and a copying process of the DNA called replication. At this point in the semester, exam 3 has already been completed and those grades should be in. So we are almost done with our entire first semester. We only have about 3 more chapters to go and only about 3 more weeks of class to take care of that. And we will be done with our first semester and then looking forward to our second semester. Hope everyone had a great holiday break, and as you're enjoying your Thanksgiving break, you might want to do the following. So far, we have covered quite a bit of information about everything from genetics all the way down to the various biomolecules and biochemistry of the cell. If you could think about the holiday of Thanksgiving and how that applies to all the various chapters that we've studied so far, it might help you in preparation for your final exam. For example, if you would think about, say, all the different foods that you ate over Thanksgiving and the different types of biomolecules, the macromolecules that they're made up of, and all the various chemistry that we talked about, and then how that gets broken down in the cell. We could talk about the respiration process, the photosynthesis process that built a lot of the food that you ate over the holidays. And so it might be kind of an interesting way to review, to think about everything that we've studied going through each chapter, 1 all the way through this current chapter 16, and how does that relate to something like the Thanksgiving holidays, from the food that you ate to the way your body reacts to perhaps the fact that if you're like most people, you gained a couple of pounds over the holidays, and how are you going to get rid of that weight if you exercise, and how does your body react to all of those situations. So if you can take the information we've learned and apply it to an everyday situation, it sometimes helps to kind of process that information and see it in a new light. This particular chapter is over one particular part of our body called DNA, and we'll get started right now. Recall way back in the early chapters we talked about those large macromolecules that are very important to biology, and one of those that we discussed, the last one we discussed, was called nucleic acids. DNA, deoxyribonucleic acid, is made up of nucleic acids. Remember that nucleic acids are polymers, long-chain macromolecules, these large biological molecules essential for all known forms of life. And nucleic acids, which include DNA, we're going to talk about primarily in this chapter. That's deoxyribonucleic acid, and RNA, ribonucleic acid, which we'll discuss a little bit in this chapter and more in the next chapter. These are all made from monomers. Remember, monomers are like the links in the long chain, and these monomers are known as nucleotides. So these are nucleic acids, which are large chains of macromolecules, and the individual links are called nucleotides. Those are the individual monomers that make up the large chain. So the overview in this chapter right up here talks about what occurred in the 50s. In 1953, James Watson and Francis Crick, you've probably heard those names, Watson and Crick, introduced an elegant double helical model for the structure of DNA. We're going to talk about the history a little bit about how they came up with this particular model and what was involved. DNA, we know, is a substance of inheritance. It's the most celebrated molecule of our time, perhaps the most important molecule in the entire study of biology, or certainly within the human body. And we know that hereditary information is encoded in that DNA and then reproduced in all cells of the body. So every cell within your body, from the single skin cell on your pinky finger to the muscle cells of your heart to the bone cells of your arm bones, all contain that set of DNA, which is the genetic instructions basically to create you. This DNA program directs the development of the biochemical and anatomical, physiological, even some of the behaviors of the living creature are dictated by this DNA. Here's a great shot taken from the 50s of Watson and Crick. Here's Watson on the left and Crick on the right. And they're looking at a wire model, basically, of their DNA molecule that they helped develop. And there's an interesting TED Talk. You might, if you're interested at all in the history of this further than what we're going to discuss, there's an interesting TED Talk about the discovery of the DNA structure and form that goes into a little bit more detail. And it talks about how Watson and Crick very much came at this from a model building perspective. So they used a couple different tools to come up with their model, and they weren't the only ones. There was almost basically a race to develop and find what was the physical structure of this DNA molecule. Watson and Crick just happened to be the ones that hit upon the right structure and the correct model at the right time. So let's get started with concept 16.1. It says DNA is the genetic material. So first let's get some background information. Early in the 20th century, the identification of molecules of inheritance loomed as a major challenge to biologists. So even after we began to understand a little bit about genetics and how this genetic material was passed on, even after we understood a little bit about what we learned from Gregor Mendel and his experiments, we didn't really understand what molecule or what chemical was the actual material that carried this information from one to another. Even if we could recognize, and by this time they could recognize that the cells had chromosomes, and they potentially understood that chromosomes were somehow involved, no one really understood exactly what DNA was or what the purpose of the DNA was, or if it was DNA that carried this information. You're going to find out that DNA is a nucleic acid, and for a long time it was thought that another type of molecule, proteins, that proteins were the ones that carry genetic information, just because proteins can have a very elaborate structure and they can be very complex. And so for the most part, many scientists felt that it was proteins that were the actual genetic information carriers. And so we'll discuss a little bit of history as how that was found not to be true. Think back to chapter 15 where we were talking about T.H. Morgan and his fly room and all the experiments he did with the fruit flies. So T.H. Morgan's group showed that the genes, the information for the genetics we're talking about, the hereditary information, is located on the chromosomes. And they knew they had seen the physical chromosomes themselves within some of the microscopy work that they had done, but they weren't sure exactly how the process worked or what part of the chromosomes carried the information. So after T.H. Morgan's group showed that the genes were located on chromosomes, then the two components of the chromosomes, which would be DNA and proteins, those became the candidates for the genetic material. So now the question was, what part of this material? Was it the DNA, which is a nucleic acid, or was it the various proteins involved? Which of those actually carried the information onto the offspring? The key factor in determining the genetic material was choosing an appropriate experimental organism. So what you needed was a creature, much like T.H. Morgan used his fruit flies. You needed an experimental organism that you could use easily in the laboratory and study this whole DNA question using. Humans far too complex, and obviously we've already talked about some of the issues with using humans in laboratory experiments. So what they did was they used a very common bacteria, E. coli bacteria. So the role of DNA in heredity was first discovered by studying both bacteria and something even more simple or more removed from something like a human, viruses that infect bacteria. It's hard to imagine that there are bacteria that can get sick, basically, and that there are viruses that affect bacteria, just like there are viruses that infect human beings as well. So this was the creature, the E. coli bacteria, and the various viruses that might attack the E. coli. This was the creature that was used to study and answer this question about DNA. And again, at first it was thought that the proteins were probably the most likely carriers of genetic inheritance because they can be very complex and they can be very different. So that made it a good candidate for carrying complex information. Plus, not much was known about nucleic acids at the time, so they were considered perhaps least likely to be the answer to this particular question. Some of the first evidence that DNA was potentially the molecule that carried this information was that DNA could transform bacteria. There was an experiment done by a British medical officer named Frederick Griffith back in 1928, and he discovered some of the first knowledge of the genetic role of DNA. He was working to develop a vaccine against pneumonia. So he was working with two strains of a bacterium, one of them pathogenic and one of them harmless. The pneumonia pathogen was Streptococcus pneumoniae. Pathogenic means disease-causing. So he had two different varieties of this bacteria. One of them would cause the disease in mammals and humans, and one of them was basically harmless. It wouldn't cause the disease. And what he found was, through his experiments, when he used heat to kill the pathogenic variety of the bacteria. And then he blended those remains, basically, and he added them to the non-pathogenic, the harmless strain. He saw something very interesting occur. Some of the living cells that were harmless suddenly became pathogenic. And when those once harmless cells were injected into mice or other lab animals, it would make them sick. So he took the heat-killed remains of the pathogenic strain and combined them with the living cells of the harmless strain, and somehow that changed or modified the living cells to make them pathogenic. He didn't really understand how this occurred, but he called this phenomenon transformation. This word transformation we now use in biology to define a change in genotype and phenotype due to the assimilation or taking in of foreign DNA. So here's kind of a graphic that shows his experiment. He had these living cells, the living, we'll call them S cells. They were the pathogenic pneumonia cells. And when those were injected, it caused the death of the mouse. So these were the pathogenic disease-causing strain of the bacteria. Then he had the non-pathogenic, the harmless version of those cells. When they were injected, the mouse remained healthy, so nothing happened. Then he took the pathogenic cells, the ones that were right here, and he killed them with heat, basically. So you may be boiling the water or putting the bacteria in an oven to where it basically causes them to no longer be alive, to kill them off. When he injected those dead pathogenic cells, nothing happened. The mouse remained healthy. But when he mixed the dead pathogenic cells with the living harmless cells, then when he injected that mixture, the mouse died. So somehow the R cells, the harmless one, had taken on the characteristics of the S cells. And when he looked in the mouse's body, he could find living S cells. So somehow the R cells were changed into pathogenic S cells. And then those S cells would reproduce and produce more of the pathogenic S cells. So he didn't really understand what had happened, but he did understand that somewhere there was material coming from the pathogenic cells that took over or changed the harmless ones and also caused them to be deadly to the mouse. Griffith's work with the pneumonia bacteria eventually led to basically a 14-year effort by an American bacteriologist named Oswald Avery, and he worked with two other people, Maitland McCarty and Colin McLeod here. And in 1944, they announced that the transforming substance that Griffith didn't really understand what was causing the change, that transforming substance was DNA. The way that they did this was some experiments in which they inactivated one of the top three possibilities every time they did a test. So you had basically three possibilities, according to Oswald Avery and his group. This material could be either DNA that was causing the change in the bacteria cells, or it could be another nucleic acid, RNA, ribonucleic acid, or it could be the proteins. So what they did was they injected material and they used chemical process to inactivate one of these three possibilities every time. So their conclusion was based on this experimental evidence that only the DNA, when it was the one that was active, that was the material that transformed the harmless bacteria into the pathogenic bacteria. So they announced this in 1944, and although it was taken note of, a lot of biologists still were fairly skeptical, and it wasn't like their information was greeted with just a major aha, this is the answer. There were still a lot of people that didn't really think that they had gotten the information right, mainly because so little was known about this nucleic acid DNA, and it was still thought by many biologists that proteins were still a more likely candidate for the genetic material. So oftentimes in science, even when a great discovery is made, it doesn't mean that overnight that information is accepted. That's why we do follow-up experiments, and you need repeatability, and you need other people to confirm your information. That's when, especially when something big like this occurs, that's when that information finally begins to be digested and accepted and then proven to be true by numerous sources. So Oswald Avery and his group, using their experimentation method, came up with their answer, which was DNA. Not RNA, not proteins, only DNA. That's the one that caused the transformation that we were looking at in this Streptococcus bacteria that made them go from harmless to pathogenic. Additional evidence that DNA was the genetic material or genetic inheritance molecule came from the study of viruses. And again, it's difficult to believe that there are bacteria that are attacked by something even smaller, viruses, but that is the case. So more evidence for DNA as the genetic material came from the study of viruses, in particular viruses that affect or infect bacteria. So these viruses are called bacteriophages, or we just shorten the word and just call it phages. So a bacteriophage is a virus that attacks bacteria alone. So you wouldn't expect a bacteriophage to make you sick, but they do infect and make, I guess you could say, make sick particular molecules, particular bacteria. So bacteriophage is a virus that attacks and infects a bacteria. They're widely used in molecular genetics research. The bacteriophage virus that's most often studied is called the T2 virus, and here's a great microscopic shot of three of the viruses right here. And they're very interesting looking little creatures, and this would be the bacterial cell that you see, and here they are injecting their DNA. The T2 virus is oftentimes studied for genetic study because there's not a lot to a virus. A virus is basically a section of DNA or sometimes even RNA, and it's enclosed by a protective coat of protein. So there's virtually nothing there. It's very simple. It's not nearly as complex as a cell. To produce more viruses, you wouldn't really say, or most people wouldn't call the virus alive because it can't do the things that a living creature can do without hijacking another cell. So for the virus to reproduce itself, what it does is basically infect a cell, hijack that cell, take it over, and use the mechanisms of that cell to create more of itself. So it doesn't have its own metabolic machinery. It basically is a protein coat with a DNA strand inside, and it injects its DNA into a cell, and then that DNA from the phage takes over the cell and creates kind of like a zombie cell that obeys the commands of the phage DNA. And the phage DNA will take over all of the various organelles of the cell and create more DNA and more phage parts, and then that cell will eventually lyse. It will burst open, and all of the newly created viruses will then escape and go on to infect other cells. Here's a great little animation of the T2 reproductive cycle. So you can right click and hit play on this, and it basically shows how the phage comes into contact with the cell, injects its DNA, destroys the DNA of the bacteria cell itself, and then uses the phage DNA to take over the cell and reproduce more viruses until the cell basically bursts open and then produces all those new viruses that go off to reinfect other cells. So because of the simplicity of this bacteriophage virus, you're basically talking about only two things. You have DNA and a protein shell. So in 1952, two scientists, Alfred Hershey and Martha Chase, they were performing experiments, and their experiments showed that the DNA is the genetic material of a phage that we call T2, a particular type of phage. And to determine this, they designed an experiment showing that only one of the two parts of the T2 phage enters the E. coli bacteria during infection. So remember in the little video clip you may have just watched, the protein shell doesn't actually enter the cell, only the DNA enters the cell. So by looking at this virus, which is really only made up of two things, protein and DNA, and determining which part of that virus actually infected and got into the cell and changed it, that kind of determined for them, well, that tells you what is the carrier of the genetic information. It's not the protein, because that protein shell never even entered the cell. It must be the DNA molecule, a nucleic acid known as DNA. That's what was injected into the bacteria cell, takes it over, and causes the whole process of viral infection. So they concluded that the injected DNA of the phage was what was providing the genetic information. And the way they do this was they used radioactive isotopes of sulfur to tag the protein, because the protein shell is the proteins that make up the viral shell is very high in sulfur. So they used a particular radioactive isotope of sulfur that the virus was using to make the shell, and they used a radioactive phosphorus to tag the DNA. DNA is a nucleic acid that's heavy in phosphorus. So then they could track what part was injected into the bacteria. Was it the radioactive phosphorus tag or was it the radioactive sulfur tag? And that's how they knew, was it the DNA or the protein that got into the bacteria and caused the viral infection. This is a great little animated clip that kind of summarizes the Hershey and Chase experiment. So if you want to right-click and hit play, you'll get some further information about how they used bacteriophages, viruses, to determine that it was DNA that was actually the carrier of genetic information. I highly recommend you take a look at this animation. It really clearly explains the Hershey-Chase experiment and how they determined that DNA was a hereditary molecule, but we'll also take a look at an illustration. We'll kind of talk about it a little bit further as well. So here's what they did. They basically had two sets of radioactive isotopes that they were using with these various viruses. So one batch was grown in a medium that was high in radioactive sulfur, and that would be the batch that would incorporate the sulfur into the protein shell. So the phages would have a protein shell that was high in this radioactive isotope of sulfur. And the other batch was grown in a material that was high in radioactive phosphorus, and it was known that the DNA molecule, the nucleic acid, was high in phosphorus. So these phages would have the normal protein shell, but they would have radioactive DNA, a DNA that was high in phosphorus that was made up of this radioactive isotope. So you would basically have two different phages. You would have one with a radioactive protein shell, and you would have one with the radioactive DNA. The next step was to allow those various phages to infect E. coli and then allow that process to continue for quite some time. And then they would take that material of the infected E. coli with either the phages that had the radioactive shell or the phages that had the radioactive DNA, and they would basically put them into a blender and blend all that material up. Then they took the contents and they put it into a centrifuge. And if you'll remember, a centrifuge is basically just a spinning device that spins the material at high speed. So the heavier particles and the more dense material falls down toward the bottom, and the lighter material collects in the top. In this case, the protein shell of the phage would be lighter and it should collect toward the top, and the cell contents and the DNA material should collect down toward the bottom. So in this batch, remember, they had the radioactive shells, the radioactive protein shells. And when they spun that down, what they found was all the radioactive material was in the top, where the protein shells were, and none was in the cell itself, the bacteria cell and the contents down here. So the bacteria cell did not contain the radioactivity. In this case, remember, we had radioactivity in the DNA. And so in this case, after they smashed it up in the blender, ran it through the centrifuge, the top portion, which was the light portion, was not radioactive. That was all the shells. But the bottom portion, which contained the cell material from the E. coli, was radioactive. And what this showed was that it was the DNA that was being injected into the cell. Out here, we learned that the protein shell did not enter into the cell, and that's why the radioactivity wasn't down here with the cell material. The DNA did enter the cell, however, and that's why the radioactive material was down here with all of the cell components from the bacteria cell. The other thing that they noticed was when they had radioactive DNA, the infected cells, the infected bacteria cells, produced viruses that were also radioactive. Up here, the radioactive protein shell viruses, when they produced new viruses, the new ones were not radioactive. And that would also tell you that it was the DNA that was passing on the information. So when radioactive DNA was passed on, well, the new viruses were also radioactive. But when the protein shell was radioactive, that didn't get passed on. And the DNA was not radioactive up here, so the new viruses were not radioactive. So basically, the Hershey-Chase experiment was a very good, well-created experiment that helped strengthen this idea and prove that DNA was the molecule that carried genetic information, not proteins. Further evidence that helped bolster this idea that it was the nucleic acid DNA that carried the genetic information was done by a biochemist in 1950 named Erwin Chargaff. Erwin Chargaff helped developed some rules that we'll talk about in just a second. By this time, by the 1950s, it was known that DNA is a polymer, a long chain molecule made up of nucleotides. And each one of these nucleotides consists of what we call a nitrogenous base, and then a 5-carbon ring sugar, and a phosphate group. And the different nitrogenous bases could be adenine, thymine, guanine, or cytosine. And you've probably seen these words and letters before. So DNA is basically a long chain molecule that's made up of a nitrogenous base, and that base is going to either be adenine, thymine, guanine, or cytosine. And then it's also connected to a sugar, a pentose ring, 5-carbon ring sugar, and a phosphate group. So we're also going to talk a lot about using the letters here. So adenine is A, thymine T, guanine G, cytosine C. Well, in 1950, Erwin Chargaff reported that DNA composition varies from one species to the next. So he collected and examined the DNA from different species, and what he found was they didn't all have the same amount of each one of these 4 nitrogenous bases. So, for example, the percentage of any particular nucleotide base was different from species to species. So you could look at, say, humans as compared to E. coli, and if you were looking at just the adenine, you might find in humans it made up 30.3% of all the nitrogenous base from the DNA was adenine. But in E. coli, it was 26%. So there were differences from one species to the next as far as how much of each of this base made up the DNA. Well, that was important because this seemed to be evidence for the diversity of DNA, that DNA could be different in the different species, and that helped make it a more credible candidate for carrying genetic material. If they had found that in every species the amount of adenine was the same and the amount of guanine was the same, that would indicate perhaps that the DNA was not the material, because why would it not be different for all the different species? But it turns out it is. So if you want to compare, say, humans to E. coli bacteria, and you want to look at what percentage of their DNA is made up of adenine, you'll find that that percentage is not the same in the different species. So this led to basically 2 rules that we call Chargaff's rules, and we'll take a look at those next. The first of his rules we already talked about, that the base composition of DNA, basically which particular nitrogenous base are you looking at, that varies between the species. So if you compare the DNA of a bird, a mockingbird, to the DNA of a human, to the DNA of a blue whale, you wouldn't get the same amount or percentage of each of the 4 bases. So that was one of his findings, one of his rules. And the second one was also very interesting. He said in any particular species, so you could be looking at, say, mockingbirds, the number of A and T bases are equal, or the percentage of the A and T, the adenine and thymine, are the same. And the number or percentage of the G and C bases are the same as well. He didn't know why, but that's what he found. If a species had, say, 32% adenine, it also had 32% thymine. So the A and T, it seemed always to be equal, and the G and C always to be equal. So that was the second of his rules that he kind of determined through his experimentation. Again, he didn't really know why that was the case, and we didn't really find out until Watson and Crick and the discovery of the double helix. But it was understood that A and T somehow matched or went together, and C and G somehow matched or went together as well. So by this point, there seemed to be mounting evidence, scientific evidence, that DNA was the molecule that we should be looking at. It's based on all the Hershey and Chase experiment and Chargaff's experiments and some of the other information we've already learned. It seemed like proteins were out, that those were not, that the protein material was not the material that we should be looking at, that it was this nucleic acid called DNA. So at that point, most of the focus began to be on DNA, and we wanted to figure out more about it. How did it work? What was its structure? What was its molecular makeup? How did it come together? And so then began basically a race of scientists around the world to try to figure out the structure of DNA, to develop a model to further understand this DNA molecule. Here's a great animation. I highly suggest you right-click and hit play. It talks about 2 nucleic acids that are very important, DNA, which is the one we're discussing now, and another one called RNA, which we will discuss a little bit more in depth in the next chapter. Both of them very important in cellular biology and pretty much very similar, except for a couple of different things that we'll discuss in further detail later. But it might be good to go ahead and right-click and hit play and watch this animation so you have a better understanding of the structure of the DNA molecule, and then we'll continue to talk about it as we go on down through our PowerPoint here. So the animation talked a little bit about the structure of the DNA molecule, and we know that it is basically a long chain made up, it's called a polynucleotide, and so it's made up of all of these particular monomers, these nucleic acids, and we know that these nucleic acids have a nitrogenous base that either could be thymine, adenine, cytosine, or guanine, and that's going to be connected to a sugar, a deoxyribose sugar, and then that's connected to a phosphate group. So see this little dotted line here? This one molecule that you see right here, that is a DNA nucleotide. So this all together represents a DNA nucleotide. And the DNA molecule is basically a long chain of these DNA nucleotides strung together. So every DNA nucleotide is made up of these three parts: the nitrogenous base, which can be of the 4 different varieties here, then the sugar molecule, and the phosphate group. And you'll notice they are connected together in a particular way. On the sugar molecule, there are 5 carbons. 1, 2, 3, 4, 5. And we name those carbons, or we number those carbons, starting with this one that's connected to the nitrogenous base. That's called carbon number 1, or 1 prime. And then we go around the ring: 2 prime, 3 prime, 4 prime, and 5 prime. And the 5 prime carbon is the one connected to the phosphate group. So we have kind of a directionality we could talk about. If we look at this chain right here, this would be the 5 prime end, because the last part of it is connected to the 5 prime carbon, the phosphate here. This portion in light blue is called the sugar phosphate backbone. It doesn't change. You'll notice it's pretty much all the same. So this portion is called the sugar phosphate backbone, and then this portion that can change is the nitrogenous base. So if we look at this molecule, we would say that this is the 5 prime end, and this is going down to the 3 prime end. So we have what we call directionality. We wouldn't see this running in the opposite direction unless it was flipped, and that becomes very important because we find out in DNA that there is another side, and it is flipped, reversed. So here we have the 5 prime to the 3 prime direction. So again, looking at the molecule, this would be the 5 prime end. That's where it ends right here, is on the 5 prime carbon. And the reason we call this one the 3 prime end is if we were to continue this chain, it would connect from that 3 prime to the next nucleotide at its 5 prime. So we have a connection here that would go from the 3 to the next 5. So because it stops right there, our open link, you might want to think of it, is the 3 prime here, and the last link is the 5 prime up here. So looking at it in a direction-wise, going from the 5 to the 3 prime end. So once scientists were finally fairly convinced that DNA was the genetic material that they were after, the next challenge was to try to determine what this molecule actually looked like. What was the structure that would help us understand a little bit further about how it worked as far as carrying on genetic information from parents to offspring. So by the early 1950s, we had a fairly good understanding of how the bonds worked in a nucleic acid chain or a nucleic acid polymer, but we didn't really know what was the exact three-dimensional shape or makeup of the molecule. So that was the next big race. There was basically a race amongst a number of scientists to try to determine what was the best model for this three-dimensional shape that they were trying to find. And some of the scientists working on this problem included Linus Pauling, a very well-known scientist who was working at the California Institute of Technology, and there were some other scientists, Maurice Wilkins and his assistant named Rosalind Franklin, and they were working at King's College in London. And so these were two groups that were hard at work, and they did discover quite a bit of information. But the scientists who actually came up with the three-dimensional model that we all know now were two Americans named James Watson. Sorry, an American and an Englishman. The American was James Watson, and the Englishman was Francis Crick. And they were not very well known at the time, but obviously once their model became accepted, they became very well known. One of the ways of studying this whole puzzle was to use a process called X-ray crystallography, and it was Maurice Wilkins and Rosalind Franklin who were basically kind of the experts of the time using this method of X-ray crystallography. That's a hard word to say. Especially Rosalind Franklin. She was well known for her ability to create these X-rays that were helpful in determining the structure of the molecule. And I don't want you to think that X-ray crystallography produced like a photograph or a picture. It produced basically what looks like, to me, spots and smudges, but this was the diffraction of X-rays off of the molecule. And from that diffraction of X-rays, you could, if you understood what you were looking at, get a sense of what was the shape of the molecule that caused the diffraction of those X-rays. So I'll show you a picture, and you're going to think, wow, how did they get a double helix out of this? But Rosalind Franklin kind of knew what she was doing, and she helped lead Francis Crick and James Watson toward this discovery. So Franklin produced a picture of the DNA molecule using this particular technique. Here's Franklin on the left, and here is one of her X-ray diffraction photographs that led Watson and Crick to come up with their model of the double helix. So Rosalind Franklin's X-rays, they helped enable Watson to deduce or come up with this idea that the DNA was helical. In other words, the two sides kind of twisted around as they went up or out. And the X-ray image also helped Watson and Crick to determine kind of the basic width of the helix and the spacing of the nitrogenous bases that they were looking at. The pattern in the photo suggested that the DNA was made up of two different strands forming this twisting ladder that we now call a double helix. Watson and Crick went on to publish a paper in April of 1953 basically describing their double helix model, and that became the most well-known model and the most accepted model for DNA. And then further investigation, further experimentation kind of confirmed that they did have the correct model. So in 1962, Watson and Crick and Maurice Wilkins as well, they were awarded the Nobel Prize for their work for discovering the molecule DNA, the model, the three-dimensional model of that. Very sadly, Rosalind Franklin, she did not receive the Nobel Prize in 1962 because she had died at the age of 38 in 1958, and the Nobel Prize is not awarded posthumously after someone's death, so she did not receive the Nobel Prize for her very important work. And sadly, she was diagnosed with a form of cancer that actually led to her death, and many people believe, much like Marie Curie, who also was a Nobel Prize winning female scientist, she also died from various radioactive poisonings, that a lot of people believe that this particular scientist as well, that she died, Rosalind Franklin, due to cancer caused by some of her work with radioactive materials in the laboratory and things like that. So 3 out of the 4 main people, Watson and Crick and Wilkins, were awarded the Nobel Prize, but Rosalind Franklin, again, had passed away before they awarded that prize, and so she was ineligible to receive that prize. You can hit right click and play. There's a great animation here that kind of talks about the double helix structure, kind of shows you a couple of different models, and then we're going to go on and talk a little bit more about the structure itself before we take a break. Here's figure 16.7. You can find this in your textbook. Let's see, I believe that is on page 309. So take a look on page 309. You'll find basically 3 models of the DNA. So here you have some of the basic key features and showing some various measurements as well as the double helical structure. Here you have a partial chemical structure model that kind of shows the bonds between the nitrogenous bases and the different phosphate sugar backbones running in opposite directions. And then here you have a space filling model that kind of shows you more of a 3-dimensional. If you could actually see it with your eyes, perhaps it would look a bit more like this on the right-hand side. So to get at their model, Watson and Crick basically were going off of the information that was coming from Rosalind Franklin's X-rays and from what they knew about chemistry and how the chemical bonds would work within the various nucleic acid chains. So they tried to build a model that would match all of this information. And Rosalind Franklin had already concluded that there must be 2 outer sugar phosphate backbones with the nitrogenous bases paired in the middle, or paired toward the center of the molecule. So using that information, they basically kind of had an insight, or Watson had an insight using Franklin's information and using her X-rays and using the other chemistry information that they knew. He had an insight that perhaps these two outer backbones, they were anti-parallel, which means they ran parallel to each other but in the opposite direction. So when they came across this idea, then everything kind of fell into place. And so the two outer sugar phosphate backbones do run parallel to each other, but they run in opposite directions, which we'll look at in just a second. And then the nitrogenous bases are connected in the center, and this forms this twisted ladder shape that we call the double helix. Their model wasn't all perfectly smooth sailing because at first Watson and Crick thought that the bases would pair together like with like. So adenine with adenine, cytosine with cytosine, etc. But when they paired them up this way, it didn't match the X-ray pictures that they had, because the X-ray pictures seemed to show that the model was of a particular width, that the molecule was a uniform width all the way through. And when they paired them up like to like, you would have some wide portions and some skinny portions. So adenine and guanine are purines. And remember, they have, or if you watched one of the earlier videos, they are larger molecules. They have two organic rings with the nitrogenous base. Cytosine and thymine are what we call pyrimidines, and they're slightly smaller molecules. They have the nitrogenous base with only one organic ring. So if you put the adenine with an adenine, you're going to get kind of a wider portion. And then if you put cytosine with cytosine, you're going to get kind of a thinner, skinnier portion. And so that didn't really match up with what they saw in the X-rays. So then they came up with the idea that there must be a particular kind of pairing. So you must put one of the purines with a pyrimidine. And when you do that, you end up with one side with two rings and one side with one ring, always. And that gives you the uniform shape and the uniform width that they saw in the X-ray crystallography pictures. So pairing a purine with a pyrimidine, that resulted in a model that more fit the information they had from the X-ray data. So here you can see an example. So the X-rays were showing that the molecule was of a consistent width, but if you would put purine to purine, you've got kind of a portion that's too wide. And then pyrimidine to pyrimidine was a portion that was too narrow. But if you always consistently connected a pyrimidine to a purine, and it could be swapped on the sides, but then you would get the consistent width that matched the X-rays. Further study proved to Watson and Crick that they reasoned that the pairings were very specific, and that was dictated by the base structures. And so they determined that adenine always paired only with thymine, and guanine only always paired with cytosine. And this would match both the physical picture that they were getting from the X-rays, and then it also matched or explained one of Chargaff's rules, was that the A and the T were equal, and the C and the G were equal, the amounts. So this would make sense. If adenine always paired with thymine, you would have an equal amount of those two. And if guanine always paired with cytosine, you would have an equal amount of those two. And so when they got all that information together, it's kind of like when you're building a puzzle and suddenly all the pieces begin to fall into place and you see a clear picture. Once they got all this information together and they built their 3D model, suddenly all this previous information that was known made sense, and all the pieces began to fall into place. Here's an example of that base pairing: adenine with thymine, guanine with cytosine. And that's where we're going to take a break. So by the 1950s, Watson and Crick had discovered this three-dimensional shape of DNA, and it was now known that DNA was the genetic inheritance carrier of this information, that it wasn't the proteins, that it was the DNA itself. And so further study can be done on DNA. And the next question was, how does the DNA replicate or make a copy of itself for this whole process to continue to pass on these genes? So that's what we're going to look at next. First, let's take a break, and I'll see you back here in just a little while.